we are starting the part 4 of this session. In part 4, we are going to talk about methods that are not that much invasive. If you go to PubMed and just search for fMRI, this is the number of the papers that you can see that in 2018 we had something like 40,000 papers with fMRI. So you can see that people have basically started from something like these years. If you go beyond that, it's just nothing there. So they have started from these years, and then it goes up, and now the situation that we are inside. So you can see that kind of things are moving forward. You can see a little bit of decline from 2018, 2019, and there are some reasons behind that as well. I'm not going to discuss about that right now. But it's quite interesting that how that the field was growing up, and then now it's in almost a plateau right now. And people are doing the fMRI studies with different sort of kind of intensities. Okay, let me go back to the slides. Okay, so going back to what we have been discussing about fMRI. So we had an introduction to fMRI. Now we are going to go and talk about details about that. First of all, what we are recording is about oxygen. So in fMRI, we are recording about kind of the level of oxygen, okay? And the level of oxygen is related to neural activities. But in reality, it is not just neurons. Even the astrocytes and the glial cells and the other things, they are also involved in that. We know that most of the time, kind of the highest level of energy that is consumed inside the brain is about the synapses. So most of the time, the signal that we have that is related to the oxygen is related to the level of synaptic activity. And it is kind of a really good proxy measure for neuronal activity. So the signal that we have is close to what we have in terms of the neural activities. But the other thing that is also important to mention that we have another area, it's called MR physics. How we measure a signal from MRI and then fMRI physics. It seems really overwhelming for people. It is something that you need to learn over time. So it is not something that you're supposed to learn in the first 2 years of your scientific career. So you still need to have time to discuss about physical physics, MR physics. And it's always a question in terms of, okay, you need to learn that in a level to be able to do your kind of research or clinical practice in the right way. Sometimes getting to kind of details that are not really helpful in the clinical work or in the kind of research work probably are not going to be that much kind of critical for you to learn. But it seems it is complex for everyone. So it is not. So if you see that there are complexity in terms of kind of the MR physics to learn, do not get worried at all. There are going to be there are things that you learn over something like 10 years. So you gradually learn about these topics over time. You need to expose yourself. You need to be open to listen about those things. Whenever I go to a kind of human brain mapping method, I just attend one of the kind of MR physics sessions just to expose myself about new things that people are discussing about. And over the time, you will learn about how these things are working together. Okay. Just to give you an idea about how the fMRI signal is being processed. So we discussed about a bold response. This is the bold response, okay? Bold response. We call that hemodynamic response function. So it is something like a function. It's called HRF. It started with a stimuli, and we discussed that there is something like 6 seconds of gap between that stimuli and that hemodynamic response. Then we have a task presentation. So for example, here we show people, let's say, angry faces. And then we show neutral faces here, angry faces, neutral faces. Or here we just give them a sound, and then sound again, and then sound again, and then sound again, and there is nothing in between. So we have a task paradigm, and then we multiply HRF to the task paradigm to be able to generate the model response. Why? Because this is the signal that we are expecting to have when we show these pictures, and we know that these stimuli are going to make these specific function in HRF. So we convolve this to the task paradigm and make this model. So this is what we are expecting to see. Okay, we are not expecting to see the signal in this way. We are expecting to see the signal in this way, because we know that there is an HRF that would translate the stimuli to the signal in the brain that we are expecting. But in reality, what we see, the signal is like this, because there is noise always there. And then we do analysis, and we try to remove what we call noise from the real signal. So this is the signal, this is the noise, and this is the value that shows how much this signal is detected here. So this is called the beta value. And then we can map that to a multi-dimensional space. This is not something that you are going to learn. So I just wanted to say that, okay, there are some kind of mathematical complexities that are not essential for you to learn. So just ignore this part of mathematical discussions. And then we have different ways of stimulus presentation, okay? When we want to have a stimulus presentation, fMRI stimulus presentation, there are very different methods: categorical designs, factorial designs, parametric designs. It's too early for you to learn about different ways of doing that. It's just going to be too complex right now. The only thing I was interested to say that, okay, sometimes we just give them one task and then we give them a control condition to be able to just compare them, and then we subtract these things from each other. We call this just a subtraction. So we do the subtraction between them. Or sometimes in parametric design, for example, we give them a sound in this intensity, and then we give them a sound in this intensity, and then we give them a sound and the next sound in a larger intensity. So, and then we show that we have a specific area that is getting activated with the same slope. So we call this parametric modulation or parametric design. So we just look for areas in the brain that are responding to the level of sound. So it's something like a dose response. And this is what we call the parametric design. So we can have parametric design, we can have block design. Then we can have combination of different things together we call factorial design. I'm not going to go to the details about that. But it's important for you to know that when we have fMRI, it's always to get sure that we will have an experimental paradigm to activate that specific cognitive function in a reliable and valid way. And this is not an easy thing to do. It's critically important for you to know that it's going to be complex to design a cognitive task that is activating the specific area or a specific function that you are looking for in the most efficient way, in the most reliable way, and it's in the most kind of specific or valid way. So there are always complexities for that. And then summarizing the fMRI analysis in just one single slide, it's just to make you overwhelmed. It's just to make you feel that it is really hard to understand. I really don't like to kind of teach things in such a way, just make you feel that it's too much complex to understand. It's really easy to learn, but it is not something that you can learn in one single slide. Okay, this is something that you need to learn in something like a 1-week workshop to know how fMRI time series are being processed to make, at the end of the day, a beautiful map. At the end of the day, making something like this. Okay, that is something that I'm not sure. There are definitely lots of good videos on YouTube. I can also have a workshop discussing about that. But there are specific kind of analytic kind of methods that we use to end up with a map like this. And this is a kind of a map related to the auditory processing. So it's just kind of hearing a sound without hearing a sound, just comparing them from each other. And we have activation in what we call auditory cortex, inferior colliculus. What was MGB? Anybody who remembers what was MGB? You can put that in the chat space. I don't have access to chat space here right now, but if anybody who is kind of who knows what MGB is, you can just turn on your video and then tell us what was that.
Medial geniculate body.
Exactly. So it is a part of the thalamus that is kind of receiving information and then kind of reflecting them to the anterior auditory cortex. And when we talk about auditory cortex, this is what we have in the temporal gyrus. In the temporal gyrus, we have superior temporal gyrus, middle temporal gyrus, inferior temporal gyrus. Here we have hippocampus or parahippocampal areas, or fusiform gyrus. These parts are so superior, middle, inferior, fusiform gyrus, or depends on the section that we are making from temporal gyrus. And there is a part, this part, that is something like transverse. So it is what we call auditory cortex. So this is a part of temporal cortex. There's a transverse temporal cortex, transverse cortical gyrus. It's called also Heschel gyri or Heschel gyrus. So that's another kind of part about auditory cortex and how this is going to process. So that is how we see at the end of the day a map of brain activation in what we call fMRI. Okay, and there are some other kind of interesting details about fMRI. Most of the time when we show people the stimuli and we show the stimuli again, the level of signal would go down. So there is a level of adaptation or habituation. Here, for example, when we give people a spoken sound compared to music, and if we have area 1 and then area 2, area 1 would respond like this. In the second time, there would be a decrease. Area 2, we have this almost the same level of kind of response even in the second exposure. What does it mean? It means that whatever area 1 is processing for that stimuli, spoken and music, is the same. So it seems that that area is processing the sound, not something related to the spoken kind of language or music. So this one is considered a sound 1, this one is considered a sound 2, and because they are the same, there is a habituation. Okay? But when we have an area 2, which is still responsive to the second sound, it means that there are neurons here that are related to spoken sound, and there are neurons that are related to the music, and they are all the same in the same place. So the level of adaptation is really low. So we have activation with these neurons in the first time, and then we have activation with these neurons in the second time. So there are some minor details about that as well. As I told you, whenever you get to the figures that are too detailed for an overview and introductory chapter in this book, there are probably an areas that the authors have a specific personal interest to that. So that's the reason that we have kind of some technical details about kind of these habituation responses. And I recently submitted a new paper for Neuroimage specifically on these habituation issues. There are, technically detailed issues that might not be your main problem for the next coming years. But they are also interesting to know that how these kind of different responses, even the habituation responses, would give us an information about the nature of the neurons that we have in different parts of the brain. Okay. Then we have an interesting issue, because what we have been doing before, if you see papers from kind of fMRI studies in Nature or Science in 1998, 1995. Most of the studies are like this. You are supposed to try to remember something. They tell you, okay, try to remember this. And then in the next block, you are supposed to not to try to remember something. And then they just make a contrast. They show that there is an activation in hippocampus, and they publish that in Nature or Science. So that was the quality of work in something like 25 years ago, okay? But now things are changing significantly. So that the things are quite different right now. And now what we have, but people realize that when we ask people not to do anything, the brain would not just turn it off. So when people are supposed not to do anything, or for example, they are inside the scanner, they see some pictures, some photos, and then they are supposed to see just a blank screen or a blank screen with a cross inside. Their brain would be activated. Their brain would be involved in lots of different processes. So people realize that we can use brain activations even while people are inside the scanner and are supposed to do nothing. And then they call that resting state fMRI. With using resting state fMRI, people realize that there are areas that are connected to each other, areas like what we call the executive control network, these areas. In the dorsolateral prefrontal cortex, areas in temporal, areas in parietal cortex. And there are networks that are negatively correlated to the control network. So, especially these kind of two networks. Here, you can see the activations that are related to default mode network. And most of these activations are related to executive control network, ECN. And if you just follow the time series here, you can see that whenever the default mode network is up, the executive control network is down. And whenever the executive control network is up, the default mode network is down. So they are in a sort of counterbalance to each other. And this counterbalance between executive control network and default mode network, what does it mean? What does it mean for psychiatry? We know that there are some psychiatric symptoms that are related to how these two networks are counterbalancing to each other, how we shift from one network to the other. And it's quite interesting that you can see here in the time domain that over something like this is 300 seconds, so it's something like 5 minutes. In 5 minutes, we have different episodes of higher default mode network. So, and when people are in default mode, they are doing sort of introspective processes. So they are seeing what is happening inside their body. They start to kind of remember memories that are related to self, as I told you before in the previous sessions. They start to evaluate things that are related to self, and they just stop thinking about things that are happening outside that are related to executive control. And how these things are kind of interacting with each other is a really interesting area for discussion. Okay, and then the question is, okay, if we have these brain mapping methods, can we use these brain mapping methods to see what are the areas in the brain that are involved in different processes? And it is exactly what we can do. One of the things that people are always asking, when you have, for example, when you have a patient with a tumor in the frontal area, the surgeon does not know that, okay, if there is a specific kind of tumor here in the right or left, whether the patient is processing the language on the right side or the left side. And where is the language processing, most important language processing areas, whether they are in the front part of the tumor or in the back part of the tumor, and how they need to approach the tumor based on the location of those kind of important brain processing areas. This is what we do for pedagogical planning. What we do, we give people language tasks, and then, for example, this here is a tumor, and as you can see, the tumor in the right side. The good thing is that for this specific patient, the language processing areas, most of the language processing areas are on the left side. So we know that if we remove the tumor from here, probably he or she would still keep the language, and it's going to be critically important. So that is good thing. So we know that kind of the Broca area and the Wernicke areas are on the left side. Okay. Or for this patient, you still see that the tumor is on the left side. The good thing is that the language area is on the right. So if the surgeon would remove this part, we can still have the language. Probably the emotional part of the language would be affected when we remove the tumor from this side, because the other side is involved in the emotional processing or the emotional content or emotional tone of the language. But at least it's the difference between being a neurosurgeon or psychiatrist. Being a psychiatrist, it's really important for you to get sure that people have a good mood and quality of life. When you are a neurosurgeon, probably if you have a patient who is coming out of the surgical unit and still able to talk and still able to walk, so these are considered as really, really important outcomes for them in their practice. And we can also combine different modes of activations, even different DTI imaging, for example, seeing what are the optical radiation from DTI, what are the different fasciculus, this is the corticospinal tract. So what are the different parts of the brain, even combining, let's say, fMRI with DTI data, and then based on that, just plan, okay, if we want to remove a tumor, how we need to get sure that we are not going to affect these really critical parts of the brain. And in reality, there is no part of the brain it is not critical. It depends on how you just make a trade-off between different functions. And some really cases you can see that these activations are around tumor, and it's really hard to remove them. So there are challenges for these what we call pre-surgical planning methods. Okay, just another important functional imaging method that we call PET. There's some physics behind it. I'm not going to discuss about the physics part. The good thing about PET is that we can make this G stands for glucose. The other method that we want to discuss about is what we call positron emission tomography. The good thing about this method is we can basically make a map from a specific chemical in the brain. The challenge is that chemical should be labeled with radioactive materials. So this one is fluorodeoxyglucose. So there are different, let's say, radioactive material. So it's, from the ethics point of view, it's always challenging to convince the IRB and ethical committees to give people, especially normal healthy control people, these radiochemicals and then make these maps. So without having really good clinical justifications, it's a little bit complex to do that. What people are doing right now is just combining PET with CT scans so they can have a better quality images from what is happening in the brain in terms of the metabolites or the material that we have. The worst thing about PET is really that the temporal resolution is really bad. So you cannot see any fluctuation. You just have an average signal for 10 minutes in one single map. So you cannot say that, okay, these networks are changing to each other. You do not have that level of information. What you see is basically a general overview about what you have in this. This one is a normal healthy person. Just an overview about the general metabolism of the brain. This one is the case with Alzheimer. So you can see that the quality of the metabolism is not as high as what we had before. And this one is a child. It's quite interesting that in children, the cerebellum is not fully developed yet, and it's still kind of in the process of development. So as a child is going to just grow up and learn different motor skills, then cerebellum would get more and more involved, and then you have, as you can see here, in normal healthy person, we have higher level of activation or metabolism in the cerebellum, okay? And then in specific types of Alzheimer's, for example, most of the time in Alzheimer's disease, we see that in the right and left side, we have kind of hypofunctions. There are some kind of types of Alzheimer's disease that they have the hypofunctions in one side of the brain. And there are specific forms of PET that are being developed for specific chemicals in the brain for Alzheimer's. So probably for Alzheimer's, PET is going to be something that people are going to use in the future, because we know that most of the kind of the structural changes that we can see with MRI are not very kind of sensitive, at least to changes in what we have in early Alzheimer, early stage Alzheimer. So PET probably is going to be used for that, but not very much for the cognitive neuroscience studies. Okay, so that was the first part.